So, hello everyone, welcome to the CCP4 Study Weekend. So there are three co-organisers. Unfortunately, Clemens can't be here today, but you have myself, Helen Ginn, and Nick Pearce at your service today. So essentially, we're going to be having a conference today on decision-making in macromolecular crystallography. So where to place your efforts, how to prioritize, when to use an automatic pipeline, when to actually dig in and get your hands dirty and go back to the theory. So I hope you'll enjoy the keynote today, followed by a discussion panel on these topics. And over to Nick to give a few announcements.
I basically have one small piece of housekeeping. Just so people know, there is also a quiet room just above the lobby by the registration desk. This room will also be streamed into there, so if you need a quiet place to go and enjoy the conference, that is also available. And Sam will come up and say this at the end, but there is also a poster session and dinner after this, so do hang around for that. And with that, I guess we will hand over to our chair, Elspeth Garman.
Thank you very much. It's my great honor to introduce our first speaker today, who is Patrick Reinke, and he's going to tell us, it's a rather long title, Lessons Learned from SARS-CoV2 Drug Screening Campaigns, Harvesting All Existing MX Tools Pipelines at the Time by a Collaborative Group of Users. And I hope it will become clear that this collaborative group of users was actually more than 100 people. So there must have had to be quite a lot of sheepdog and trying to herd cats to get this to work. So we're looking forward very much to hearing about the effort. Do you want to use that microphone on this one?
Thank you for the nice introduction.
Oh, sorry, I meant to say that, sorry, that he did his MSc in Göttingen, his first degree in Hanover, PhD in Hanover back, so sort of like a yo-yo, and is now in Hamburg at the CFEL. Sorry about that, Patrick.
Thank you. All right. Yeah, thanks a lot for the organizers to invite me, especially Karen and Helen who approached me. Yeah, today I talk about a Corona story, of my Corona story, what I did during the worldwide lockdown. Yeah, and how we performed a huge X-ray crystallographic screen on the SARS-CoV2 main protease. So everything. Yeah, everything yielded into this publication. This is, you can see there are a lot of authors, so it's not a work of a single person or a few person, but it's really a work of over 100 person, 103 to be exact, and everyone contributed valuable things to it. No, sorry. Laser pointer is new for me. Yeah, everything starts in the year 2020. At the 1st of January, after I finished my studies for the PhD, I started my postdoc in the workgroup of alkamenes at DESY in Hamburg. And when I was 1.5 years old as scientist, month old as scientist, we went to APS and made some nice serial crystallography experiments, and then everything started with the pandemic. There are a lot of restrictions in flying, and everything was going to lockdown soon, and we were a little bit afraid to don't get back to Germany again. But my boss Arke, he said, so we need to do something as scientists. We can contribute something. He discussed about to perform a screen with a repurposing library to accelerate the drug development. While other people just bought a lot of toilet paper and rice, then we thought about to do something against this pandemic. yeah, actually, we made it back to Germany, and in the 12th of March, Arke just got some collaboration partners. It was, of course, Henry Chapman, Christian Betzel, Arwen Pearson from Hamburg, Ashwin Chari from Göttingen, Andreas Saliani from the Fraunhofer Institute in Hamburg, who got a lot of experience with libraries, screening libraries, especially repurposing libraries, and Matthias Rarey for computational science. Yeah, you can see here that repurposing library usually are very big compounds or bigger compounds in molecular weight, more complex. But when you find a hit which binds to your target protein, then the interactions are most likely very strong, and the affinity could be bigger. Yeah, and our main target was also very clear quite soon. So the main protease of SARS-CoV2, because Rolf Filgenfeld published this structure right in the beginning of the pandemic, and he also offered to us to work together. He was willing to give us a plasmid and some protein and protein crystals for initial work. For everyone who never heard of the main protease, you can see it here. There are two entrance pathways for the virus. It's not important at the moment, but important is here. The virus injects the genome into the human cell and uses the protein production apparatus from the cell and produces a long polyprotein. And this polyprotein is not functional, the single protein parts, but this MPRO, this little heart in the middle, cuts it out itself, and then also cuts all the other protein parts into a functional form. So the invasion of the virus can start. Yeah, and 23rd of March, we received a plasmid from Rolf Filgenfeld, and we started protein production. And then Arke came to me and said, you are a scientist now 3 months old nearly, so you take over the crystallization of this huge project. And at this point, we were like 50 to 80 people, and it was a huge pressure, but also a very nice decision to take this opportunity to show that we can perform this kind of screening. I tried a lot with the condition. Our goal was to get a pH in the crystallization condition near to the physiological pH, so the binding of the compound has the same pH as the cell would have, and also like there must be some DMSO into there or some solvent who resolves all the compounds, because usually screening compounds or drugs are not very soluble, so we needed some DMSO in this case in there. And yeah, so I was able to crystallize with 5% DMSO, which is quite high, but MPRO is the main protease is very tolerable in this case. Yeah, and yeah, 2nd of April already, we were able to measure first crystals at P11 from the screen. The exact timeline is here. We collected alone in April, which was the main time frame of the screen, over 7,000 data sets, and generated 41 terabyte of raw data. And yeah, it all begins in the 2nd of March, or 1st of March, and with 400 data sets a day. And very fast, we got our first hit. It was around Easter, so my boss always called it our Easter egg. It was a first nice motivation to go on and increase the speed. So the ample beamlines joined also shortly after this, and in the end, we were about a throughput of 600 data sets a day. Our general workflow was like this. You can see that the protein production is, of course, the first part. We were a lot of work groups which collaborated in this for this was the University of Hamburg, Christian Betzel, Armin Pearson's lab contributed. CFIL was Henry Chapman, and also the XBI from European XFIL, the sample preparation lab, produced a lot of protein. All the protein went to me, and I were able to do a lot of crystallization. So the Fraunhofer Institute had an echo system, a nanoliter dispenser. They pre-coated the crystallization plates. Then I dried it under vacuum, and then I applied the crystallization solution on the solid compounds. For this, I chose the Oryx robot because we also make seeds, seeded the crystallization, the crystallization setup. To get a very reproducible result in the crystals. Yeah, this took the Oryx very accurate, but also very slow. So I sat night by night in the laboratory alone, but everyone was at home, so there was nothing better to do. And yeah, in the end, it was a nice experience. Yeah. After that, after the proteins, the crystals formed, and they needed to be harvested. For this harvesting step, we built 4 harvesting stations with a microscope and with a lot of loops. So we collected all the loops from the campus and from where we can get it, and ended up in some wild mixture of loops, like nylon loops, big loops, thin loops, barrel-like loops. So in the end, it was like when you harvested the crystals, and you pick the loop, so a loop, then you never know. It was like a gamble. Is this a nice loop you like, or the bad loop? So from this, we learned, of course, in our further development of this crystallization screening facility, we have one type of loop, one type of pin, which is QR coded. So you can also, you are able to track it when you scan it in. So yeah, this was one messy thing, which was a downside in the harvesting. But yeah, then the crystals went down to the beamline. So every 8 to 10 hours, we needed to feed the beamline with new packs. So like 24 packs we were able to load. And then people measured mainly, so they measured remotely, mainly from Hamburg, but also one person in San Francisco who did night shifts, or also in Moscow. So it was some worldwide collaboration at this point. And then, of course, you need to track every crystal. So for every crystal, we filled in a table of form. So this was our first approach, and there's only a small amount. So in the end, we had like 20 centimeter pile of sheets with the crystals. At this point, we just went on and on, and in this case, Armin Pearson just said, we need to break a little bit. We need to develop a database to make everything nicely trackable. And so on the fly, Martin Damraki and Luca Galizio from CFIL, from Henry Chapman's group, developed a nice database where we can track every crystal and see which compound is in it. Yeah, and this database is also further developed until today. So we use it today still. And now it's web-based, and you can fill everything in from the plasmid really until to the final hit refinement. This is very nice, and it's very easy to handle. This is also one thing I won't miss anymore, that you have a big screening campaign, and you search for one compound and all the crystals you've harvested for it. And with this database, it's quite nice, and like additional information, like resolution and based on CC half, for example, or R3 I chose here. And also it goes really to the end of the pipeline. So with indexing, you can nicely choose programs which you want to use, and also for model building. Which data sets do you want to use? If your data is anisotropic, you can use star aniso data and so on. So this is really a thing which is very helpful. Yeah, and then after, or back to 2020, after we collected all the data also needed to be processed. This mainly did Alexander Yafanov from CIFO, from the Chapman Group. He used in the beginning XDS modified version, and after, later on Autoproc, where XDS, aimless X-scale pointers and star aniso was included. And also later TJ Lane, also young group leader in CIFO and Chapman Group, joined, and they also used dials. And then we are now somewhere between April and May. Then the first hits appeared, and the hit finding was automated. So we first built some models. For this, TJ Lane and Dominic Overture used Dimpo and AutoBuild. And luckily, Helen Jin also joined our collaboration, and she is the author of Cluster4X. And with Cluster4X, we made some homogeneous subsets, which increases the chances of hit chance for further analysis. Yeah, and at first, Helen found 63 clusters, and TJ later on also tried a little bit to play around with this with Cluster4X, and he found 7 clusters, or formed 7 clusters. And then we had overall 43 hits. And then my job, I came back into the game together with Sebastian, Julia, Wiebke, Dujan, and Dominic. We refined a lot. So, and this was one point where the project slowed down a little bit. So when you can see here in the timeline on the right, the final refined hits were at September around. The problem here in this case was that we use repurposing libraries and the compounds. And these repurposing compounds are very complex, and they do also some kind of chemistry in the crystallization drop. So a few hits were very difficult to model, and we saw that some of the compounds are in reality not complete drugs, but pro-drugs. So they cleaved some part to release a warhead. And yeah, that was in parts also not known before. So this made everything a little bit difficult and slowed down everything. Yeah, you can see here some nice hits. So most of the hits were located in the active site of the main protease. When you turn it around in the back side, you can see also some cluster. And this 2 cluster were quite interesting, and we proposed that these could be allosteric sites. Yeah, but as soon as we had some the hits ready, we sent the compounds to the Bernhard Nocht Institute, also in Hamburg, to the group of Stefan Günther, his postdoc Yaisa Fernandez Garcia. She made some antiviral assays on cell level. So she used various 6 cells to see if the compounds we delivered had antiviral activity. And yes, so overall 7 compounds showed antiviral activity. One interesting compound is here, shown in yellow. This is AT7519. The antiviral activity is quite moderate. So the EC50 is 25 micromolar. But a few weeks ago, before this, El Baba et al. Published the green compound here. and they estimated that this inhibits the dimerization, thus infecting the activity of the MPRO. And we found, so they proposed that this arginine 298 is crucial for this interaction, and yeah, our compound also interacts with this arginine and change the conformation. But our most promising hit was a peptidomimetic, so it mimics the natural substrate, a polypeptide of this main protease, and so here we had some nanomolar EC50. Yeah, this in principle was our main result, so finding a very nice high affine compound inhibiting the main protease or the viral activity. Everything summed up here in some numbers. Over 6,000 compounds we screened, and in the end we published 29 structures into the PDB, and yeah, with one very high affine compound. Yeah, and overall the hit rate is quite low, with 0.6%. So usually with fragments, it's higher. But of course, as a reminder, this repurposing compounds are quite complex. Yeah, to follow up this, we modified this calpeptine with a known warhead protection. And we figured out that this calpeptine is very, very affine to a human protein, that's cathepsin L. And the affinity is here picomolar, so very, very, very strong. And this S-calpeptine we tried in animal trials, and there we were able to see that in the trachea with very low dose, it inhibits the viral load, the viral reproduction. So it's active in the trachea in this case. Yeah, and here you can see the mechanism. So the cathepsin L is in one pathway, the endosomal pathway, a crucial protein which activates the virus and leads to the fusion with the membrane, and which yields to the release of the genome. So, and this with this drug we can not only inhibit this pathway but also both pathways by inhibiting the MPRO2. Yeah, in the end, I really need to say thank you that I was able to be a part of this very nice but big collaboration. Yeah, everyone was valuable in this contribution, had valuable contributions, especially thanks also to Sebastian, my colleague, who I get a lot of feedback and inspiration, and Alkmains, my boss, to give me this big, huge task, which I really liked. And as a reminder, I was 3-month-old postdoc in this case. Yeah, we went on with screening. So we established a screening facility at DESY. So we do screenings on an everyday basis. We had some more corona-related screens, but also now switched to other targets. One thing what we could make better retrospectively. So the social media communication was not existing a lot with our group. So the Diamond collaboration, who also made a nice successful screening campaign, showed how it should be. So I guess all of us scientists should be more publicly proud of the work and show also maybe on social media what they do, and so the people can see what happens in the secret laboratories, which are not secret at all, but yeah. And in the end, I will not miss like programs or platforms which offers the whole pipeline from the beginning of the protein production to the end of the hit finding. In case of the Armacord, for example. So this is one thing which is really helpful and user-friendly. If the user can easily use a platform, it accelerates the experiments. And of course, automation. So this crystallization harvesting in a fast manner is hardly to improve, but of course, data collection is nicely automatable. So this is one thing which is in Diamond already established, but we also work at our new beamline at P09 called HiFax to automate everything and keep the manual effort low as possible to accelerate everything as fast as possible. Yeah, and to feed a little bit the panel discussion, my new main project is electron diffraction. So I'm crystallographer by heart. The main goal is to crystallize, to measure some proteins soon with high energy electron diffraction. And our new source offers some very nice mega electron volt electrons and a Jungfrau detector. And we already had first nice results. So we are at the moment at measuring inorganic crystals. This is one silicate layer we measured, and yeah, very recent results are here. And with this, I want to end my talk. You can see this very nice crystal I prepared for this electron diffraction, and you can see it's usually a 5 by 5 angstrom unit cell, but you can see here a superstructure because it's nicely ordered, but this is very beautiful in my eyes, and every time I see like diffraction patterns like this, I'm reminded that why I chose this nice job. So thank you.
Thank you very much. It was an amazing effort. Do we have questions? Please, could you say who you are if you ask a question? Sorry. Any questions? Can I ask a cheeky question?
Yes.
Why did you do it in that order rather than screening first to see if they were antiviral and then finding out where they bind?
Yeah, basically there were during the pandemic a lot of screening campaigns based on mass spectrometry, on chemical assays, and we chose this because it's this route because it's very sensitive with this PanDDA analysis from developed by Reinke. Is very sensitive, and we can see also very low binder, and this could help later on with drug development to modify the drugs. But the overlap of all the screening techniques like chemical or whatever, the overlap of the found compounds, even when they use the same library, was very low, or very small. So yeah, so for this we chose this route.
Thank you. Up there, thank you. Can you say who you are, please?
I'm Ben Banks from Cardiff.
Sorry, I knew that, but...
Was that from a targeted library or from a random library? Was...
Please, what was the question?
Where did the hit come from?
This is from the Fraunhofer library, yes. So this was from this huge repurposing library.
You have more questions for Patrick? Okay. Flick.
It's Patrick from Diamond. On these big collaborations across institutions, what tools did you find particularly good to enable scientists to work well together?
Yeah, in principle, like this self-developed platform or tool or database. So this is really suited or really made for this collaboration. So for people who only use the diffraction data to analyze, or other people just use it for the measurement, and this kind of platform which offers all these sub programs was really a game changer, and it's really helpful for collaborations.
So Dave Brown from Serbia. So a really nice heroic effort, do what you can do with the tools you have. I think coming back to that question around what was valuable for the cross facility and cross user group interaction, you talked about the database, the crystallographic, but how did you combine the data from different data sources? So where did both your full database of all the different data and different techniques, especially around Antiviral, and where was that freely available from the same platform?
Yeah, so principle, the database at the moment stops at the refined hit. So further gone with this antiviral activity and so on, we so far not included it. But yes, this is also one point which is a very nice idea.
What was the actual chemistry follow-up on
yeah, so in principle, we just modified the warhead. So in principle, yes, you can elongate the peptidomimetic parts, but yeah. So in this case, we focus on the warhead because it was an aldehyde, and aldehydes usually are metabolized quite fast in the human cell.
Any more questions for Patrick before we? Oh, yes.
Thank you, Patrick, for the nice talk. I realize you used a range of compounds of different sizes, like from really small fragments to the big residue-like small molecule. Did you have any restrictions in choosing your library in terms of the size?
Actually, no. So it's more likely the smaller the compounds are, it's more likely that they bind. Usually with fragments, you always have some binder, like 10 to 15%. But we had also compounds like 5,000 kilodalton, 5,000 Dalton. So in principle, there was a limit. So 5 to 6,000 Dalton was the limit for us. But yeah, for these huge compounds, we did not observe any electron density rates.
Yeah. Do you think the calpeptine?
Calpeptine?
Yeah. Do you think it's larger than 500 kilodaltons?
What, please?
Do you think it's larger than 500 kilodaltons?
No,
Sorry, can I repeat, because the people on Slack can't actually hear the questions.
Yeah.
So,
okay.
do you think
that was because one of the
Sorry,
microphones wasn't connected, but they should all be now connected to the live stream, so we should be good now.
Sorry, please answer the question.
Yeah, it's 560 Dalton. So it was a larger one, but yeah.
Yeah, because it's quite large.
From a perspective of a fragment user, yes, it's in this case very large.
Okay, I think we'll take a last question from up there.
Thank you. Graham Winter from Diamond, but also on behalf of some others. You mentioned XGS brackets modified.
Yes.
I appreciate that may be a longer answer than we have time for, but several of us are curious as to how XGS was modified.
It was Alexander told it to me, so he is a very nice X-ray data processing guy. And he just modified it for himself, like excluding water rings. We had, so the quality of the crystals was quite varying. So some crystals were perfectly fine, and the other one, due to the fact that the harvester, the crystal harvester had some problems to get the crystals, or they were inexperienced. So there was some more like ice forming. And yeah, so we had in some cases problems with ice rings, and this was one thing he modified. That
to be clear, so XGS itself was not modified; it was how you were using it was modified.
Yeah.
Right. Thank you.
That does explain why you said that you froze the crystals rather than cooled them.
Yeah.
Whoa! Sorry, I was trained as a physicist, and to me, freezing means you change the phase.
Yeah.
Right, and you try not to when you cryocool. So you cool them. But if you got ice rings, you froze them.
Sometimes a problem.
For anyone who had that on their CCP4 bingo, you can cross that off.
I think we'll thank Patrick very much for a great start to the meeting, and thank you.